Hello, welcome to 13 Universal Learning Beyond Borders. This is Shedro, your biology teacher. So I'm going to be taking you on a ride on solving A-level CIE biology questions. Okay. Just to let you know that every question from this that we will be looking at solving in this video is from CIE past questions. Okay, we're going to be looking at cell structures series 6. So I encourage you to go to our videos and watch series 1, 2, 3, 4, 5. Okay, very educative, videos. We broke stuff down that can help you understand, right? So in this video we're going straight away. Just get set, let's explore, let's dive to solve questions on microscopy. All right, okay. The first question says the photomicrograph, you can see that, says the photomicrograph shows a bronchiole and alveoli. All right, these are the alveoli surrounding this bronchiole. Okay, all right, so let's see what it says. The magnification of the image is 3 times 360. Then I said, what is the maximum diameter of the bronchial lumen? This is the bronchial lumen from here to here. That is from here. All right, this edge to that edge. The lumen is this white part. So what is the maximum? The maximum, it will be from this side to this side. Either I said, what is the minimum? Okay, measure from here to here. Okay, all right. So normally what you're going to do if it's an exam, of course, what you do is that you use your ruler and measure from here to here. Okay, now most times, of course, if you try it here, you may not get the answer because the font has changed. But the real hard copy questions, just take your ruler, measure from here to here. And of course, if what you have is in centimeter, like when you measure this, it's going to give you about 5 centimeters. So that will give you about 5 centimeter, right? And I've always said in my previous video, you should convert centimeter to micrometer, because micrometer is the basic unit to work with in microscopy, and you do that by multiplying with 10,000. So if you're moving from micrometer to centimeter to micrometer, multiply by 10,000. So this is about 5 centimeter. So or more, this will give you 50,000. Okay, and don't forget that whenever you measure, when you measure the diagram, the photomicrograph, the measurement you get is the image size. Okay, don't forget that what we're looking for here is the actual size. Remember that magnification, all right, basically is equal to image over actual, right? And then actual will be equal to, if you make actual sort of formula, will be equal to image over magnification. Is that okay? Yeah, so we already have our image and we have our magnification. Image is 50,000, so 50,000 all over. Magnification is 360. So by the time you divide it, okay, I want you to bring out your calculator and punch this. It will give you something else, something closest to 140, right? So 140 is most likely the answer. Is that okay? Something close to it. But don't forget the step, measure the image. Okay, if it's in centimeter, convert it to micrometer. So you convert it to micrometer by multiplying by 1,000 and 10,000. So this is now a micrometer. Is that okay? If your ruler is in millimeter, all right, so you can always convert millimeter to micrometer by multiplying by 1,000. That's 10 raised to the power 3. Okay, but get it to micrometer, very important, and then you can continue the calculation. All right, so the answer, of course, is this. By the time you 50,000 divided by 360 will give you something close to this. That is the answer. All right, the micrometer. Okay, you may not compare to micrometer if the options doesn't have micrometer unit. If you ask millimeter, you leave it in millimeter. All right, after the calculation. All right, let's go to the next question. This is an interesting question, so you can pause the video and try to solve it. Okay, now the question says a specimen is observed twice. Now they observe it twice with a microscope. Firstly, using green light with a wavelength of 510 nanometer. So green light was used first, and this is the wavelength, and then using red light with a wavelength of 650 nanometer. Okay, so if you check this, then I said, where's the question? The question is just an option. Okay, the question says, what happens to the magnification? No, what happens to magnification and resolution when using red light compared to green light? So what they changed here was the magnification. What they changed here was the light. Okay, and please don't forget that this 510 nanometer, all right, is shorter than 650 nanometer. Now, the shorter the wavelength, the higher the resolution. Okay, resolution is the ability of microscope to distinguish between two separate points, but it is also a product of wavelength. So the shorter the wavelength, the higher the resolution. That means more details will be seen. So by moving it from 510 nanometers to 650 nanometers, what we have done is we have reduced the resolution. It has decreased, but magnification has remained the same. So remain the same, C and D, but we'll come to resolution. The resolution actually decreases because 510 nanometer will give us a higher resolution because it will be able to see more details than 650 nanometers. Is that okay? So for example, this can see things that are lower. All right, sorry, from 510 and above. This is structures that are from 650 and above. So you can't see anything lower than 650. So this sees more. So if you move it from here to here, you have reduced the resolution, the amount of details. So the answer is therefore C. Magnification remains the same while resolution decreases. Okay. All right. Thank you. Please, you can give us your comment, like this video, share it, and then let's go to next question. So the diagram shows a section through epithelium found in part of the respiratory system. Let's look at it. That's it. All right. So what is the magnification of the diagram? Okay. So like I said, I've always said this. What you do, remember that magnification, the formula of magnification, all right, is equal to image size over actual. So actual, right? So in this case, we are dealing with a scale bar. This is a scale bar. So the magnification formula changes, even though it implies the same thing. The magnification formula here will be length of the scale bar. How do you know it's a scale bar? This is it. This is the bar, right? Up and lower. The length of this scale bar. So take your ruler and measure from here to here. That will be the length of the scale bar, which actually represents image or size. Okay, and then you now divide it by the value on the scale bar, which is 80. This value on the 80 actually represent the actual size. So if you take your ruler, of course, and measure here, you get something close to about the real question, right? The hard question. Remember that all the measurement may not be correct if you're measuring this soft copy here, because of we've worked on this, change the font, all right, when we extracted it from CIU questions. Okay, yeah, we're using CIU questions for this, all right, and we give them credit for that. They are the source of this material we're using to teach you to solve this question. All right, now so you measure from here to here. When you do that, you have about 2.8. okay, so you have about 2.8 centimeter. Remember what I said? So that length is about 2.8 centimeter, and 2.8 centimeter, I wanted to convert it to micrometer. Remember what I taught you, that you have to convert centimeters or millimeters to micrometers. And if you are moving centimeter to micrometers, you multiply by 10 raised to power 10,000. That's 10 raised to power 4. Okay, sorry, 10 raised to power, let me clean it. Okay, okay, so that will be 10 raised to power, yeah, 10 raised to power 4. So 10,000 is 10 raised to power 4. 2.8 times 10,000 will give you 28,000 micrometers. All right, 28,000 micrometers is the size, the length of the scale bar. Okay, so you divide it by the value on of the scale bar, the value on the scale bar, which is 80 micrometers. Okay, so by the time you divide that, get your calculator and divide 28,000, all right, divided by 80. 28,000 by 80 will give you 350. Okay, so the answer is times 350. That is the magnification. Or I can pause the video or just replay this video. You can send us question if you have comment. So let's go to the next question, guys. This is very interesting. All right, I want you to pay attention here. This question 4. Let me get the options. Okay, A, B. All right. Okay, good. Look at it. It says an experiment was carried out to separate the cell structures in an animal cell. This is ultracentrifugation. Cell fractionation. Cell fractionation. The cells were broken up. The extract was filtered and put into a centrifuge tube. This tube was then spun so that the heaviest cell structure sank to the bottom first, forming pellet 1 as shown. This is pellet 1 as shown. So this is the supernatant, that's the pellet. The supernatant contains less heavier organelle. This is the heaviest one. It settles first, okay, when it's spun at a lower speed. Then of course the liquid above pellet 1 was poured into a clean, all right, centrifuge. So this liquid here is called supernatant, is drained off into another tube and then spun again at a higher speed to separate the next heaviest cell structure. This cell structure sank to the bottom, forming pellet 2. So we've got pellet 2. The same thing, the same procedure was repeated. Okay, it was repeated to obtain pellet 3 and of course pellet 4. Each contain a single cell structure. Single cell structure. So what is the function of the cell structure extracted in pellet 1? All right. So if you are used to, of course, I think I've mentioned it. Is it the first time I'm saying it here? All right. There's what we call, is an acronym. We'll call it nucleomyelin. Okay. It's an acronym known to show the different sizes of organelle, cell organelles. So we have nucleomyelin. Okay, nucleomyelin. That's all right. Now, but you see, I added something to it to accommodate plant cell. So I added C nucleomyelin. So if we're dealing with plant cell, the heaviest structure there will be cellulose. All right, this C is cellulose. For plant cells, it's going to be pellet 1. All right, this new is nucleus. Okay, that's pellet 2 if you're dealing with plant cell. This chloroplast is the next heaviest, which is pellet 3 if I'm dealing with plant cell. My is mitochondria, pellet 4. L is lysosome, pellet 5. E is endoplasmic reticulum, pellet 6. Ribosome is the smallest, the less heaviest. All right, the least heaviest, okay, which is R. But now we are dealing with animal cells, so I start from nucleus. Animals don't have cell wall, so this is pellet 1. Pellet 1 is nucleus. That's the first one that comes out. It's pellet 1, nucleus. All right. Now, if I should continue, I don't have to, but let me continue to provide more information. We cannot choose chloroplast because animal cells don't have chloroplast, but pellet 2 is going to be mitochondria. In case in a question they ask you for pellet 2, they ask you for pellet 3 in an animal cell, that will be lysosome. That's pellet 3. Don't forget that. pellet 4 is going to be E endoplasmic reticulum, that's P4, okay, and the last one here is pellet 5, which is ribosome, okay, if you are working on an animal cell, okay. So now, of course, pellet 1 is nucleus. So the question is, having said that it is nucleus, and they're asking us for the function. So what is the function of nucleus here? A says digestion of old organelles. This is for lysosome. B says production of ATP. This is for mitochondria, and of course chloroplast. They both make ATP. Now C says production of messenger RNA. This production of messenger RNA is called transcription. Transcription occurs in the nucleus. So this is one of the functions of the nucleus. But if you go to D, which says synthesis of protein, this is a function of ribosomes, or RER. So the answer, all right, is what? C. So nucleus, one of the roles of nucleus is production of messenger RNA by transcription. I hope you got this. Okay, chat us and let us know what you think. Next question. The photomicrograph shows onion cells. That's onion cells, right? The actual length of the onion cell labeled Y is 350 micrometers. Then answer, what is the magnification of photomicrograph? Okay, don't forget, guys, that they are asking you for magnification. Magnification is you know how we do it. Okay, pause this video and try solving this question. Okay, pause it and try it. Okay, so magnification is image height, you know that, over actual. Okay, and you know that image height is, we get it from measuring the photomicrograph. So you measure the photomicrograph from here to here, from this point to this point. Okay, and of course use your ruler and measure it, the hard copy. Right, if you have the hard copy, you measure it, and when you do that, now for this, I've always said you may not get the exact measurement because of the change in font, all right, of this material. But when you use the real paper, the hard copy, the exam question, all right, that Y actually, when you measure it, is about 14 centimeters, right? It's quite long, 14 cm. So I said we don't work with centimeters, convert it to micrometers. Okay, how do you convert to micrometers? All right, you multiply by 10,000 and that will give you 14,000. So image height, image size is 14,000, right? 14,000 divided by actual. Actual is 350. Okay, so get your calculator and do this. Get us the answer. What do you think is the answer? Punch your calculator. 14,000 by no, it's not 14,000. Is 140,000. Okay, yes, 14 times 10,000. Because if you are moving micrometers to sorry, centimeters to micrometers, you are multiplying by 10 raised to the power 10. Okay, 10 raised to the power, sorry, 4, 10 raised to the power 4, yeah, which is 10,000. 10,000 10 raised to the power 4. So multiply 14 by 10,000 to give you 140,000. Is that okay? Divide it by 350. Punch your calculator. 350, of course, is our actual size. That's our image size. Okay. All right. So punch it. What is the answer? The answer is 400 times 400. That's the magnification. Okay. Now let's go to the next question. It's a which range of cell diameter is typical for prokaryotic cells? Now, please, we have said that prokaryotic cells, the range is from 1 micrometer to 5. So if you check this, 1 micrometer is about 0.001 of course nanometer. So this doesn't fall in. This is that's 1000 nanometer divided by 1000. It will give you 1 micrometer to 5 micrometer. This is the answer. Okay, don't forget, it's a fact. The range of measurement cell diameters of prokaryotes is from 1 micrometer to 5 micrometer. So this nanometer, 1000 nanometer makes, of course, 1 micrometer. So you want to get micrometer from nanometer, you divide by 1000. So this gives you 1 micrometer. All right, 5 micro. This is the actual measurement for prokaryotes. So the answer is, of course, B. Right? Okay. Send us question. Let's know what you think. Now look at this. They say, what is the largest? What is the order of size of cell components? This is from the largest, right, to smallest. Anytime you see this question, be excited, right? And then bring nucleomylar, bring C, all right, nucleomylar. You know C nucleomylar, right? All right. Of course, nucleus is nu, my nucleomylar, nucleo. This is um chloroplast. So the largest is nucleus. Now, of course, cellulose is not featured here. It's the heaviest, but largest is nucleus. So we'll check the option that has nucleus, and that's B. Okay. All right. Of course, after nucleus we have chloroplast. Then we have my. My is mitochondria, and L is endoplasmic reticulum. E is rough and endoplasm. L is lysosome. Sorry, lysosome. This is endoplasmic reticulum. R is ribosome. Okay. So nuclei. So if you check what we have done so far, you can see that it goes with this nucleus, many of pluralized nuclei, chloroplast, mitochondria, ribosome is the smallest. Okay, that's R here. So that's the answer, right? So the answer is B. Okay. Now look at this. This question, so this question is not complete. We're going to solve this question later. Let's go to number 9. So what are the appropriate units, okay, for measuring diameters of alveoli diameters now and width? Now for cells, we measure cells using micrometers, tissues are measured using millimeters, and other cellular components, of course cells and larger organelles like nucleus and chloroplast are measured using micrometers. Okay, so but small other structures are measured, smaller structures are measured using nanometers. So if you check A, we don't use millimeters for cells, so that disqualifies. This micrometer, yes, but this millimeter, we don't use it. Check C, this micrometer, nanometer, this looks like the answer. This millimeter, that is eliminated, so use micrometers and nanometer. That's the answer. Okay, so the answer is C. All right, guys. Look at number 10. Which structure is measured in units shown using a light microscope? Look, we cannot use light microscope to measure cell membrane. You cannot even see cell surface membrane. It's too small to be seen by light microscope. So that eliminates this. Cell wall cannot be clearly seen using light microscope. Can measure it. Okay. Now, ribosome is too small to be seen by light microscope. The size is about 25 nanometers for 80S ribosome. All right. So you see, based on what can be seen, A has been eliminated, B has been eliminated. But you see, chloroplast is a big organelle, and I told you guys that big organelles and cells are measured using micrometers. Okay, but other small organelles use nanometers. Right. So this is why chloroplast, right, can be seen using with light microscope and is measured by the unit micrometer. So the answer is C. All right. Thank you so much. We thank you for spending time to listen to this video. You can replay it, but you know, you can leave the comments on the chat, like the video, subscribe, share. So this is where we are stopping for now for cell structure series. We're going to continue later, but next class we're going to be dealing with biological molecules series 1. Don't miss it. Okay, go on for it. We're going to be cracking down the biological molecules, looking at their structures and their properties, and of course and their roles, how the structure determines the properties and the properties determine their roles. See you next class on cell biological molecules series 1. Bye.